院长、副院长，还有各位院士，大家好。今天很高兴来介绍今天主题演讲的第一位演讲者蔡立慧院士。蔡立慧院士呢是在1960年生于台湾，他从中兴大学兽医系毕业以后，就到美国深造，先后在威斯康星大学、德州大学攻读分子医学跟技术学，博士后研究则专攻神经科学。随后任教于哈佛大学的医学院，目前是担任麻省理工学院学习与记忆研究所的讲座教授兼所长。同时，他也是美国霍华修斯医学研究所及麻省理工学院部落格中心的研究员。蔡院士主要研究的领域包括了细胞生物学、遗传学跟分子生物学。他对于阿兹海默氏症的研究贡献是享誉国际。他曾在1999年的时候，透过老鼠的实验，证明了借着环境的刺激，还有药物的治疗，能够使记忆退化状况的老鼠重启学习和记忆的能力。这个研究在当时颠覆了以往认为阿兹海默氏症是没有办法治疗的观点。当时在《Nature》杂志发表时，除了在学界投下了一颗震撼弹以外，也为阿兹海默氏症的病人开启了一串希望之窗。此后，蔡院士有很多的研究都发表在《Nature Science》或者《Neuroscience》的著名的期刊。蔡院士医生获得奖项无数，我想我没有办法念，要不他演讲就不用讲啊。然后呢，他获选为本院生命科学组的院士、美国科学促进协会的会士，还有美国国家医学院的院士的殊荣，在神经科学里面具有崇高的地位。今天我们中央研究院十分荣幸能够邀请到蔡院士，很年轻的院士来院士会议主讲这个他在神经科学领域的研究精神跟发现。那他的演讲题目是 The Promise of Neuroscience。让我们大家以热烈的掌声来欢迎蔡院士。
Today, this morning, I would like to just bring you guys some updated information about the new technology and new approaches to investigate the secret of the brain and how to better investigate neurological disorders. So today, biomedical research is advancing at a lightning speed, and many life-threatening diseases now can either be cured or brought under control with effective treatment. However, we have not achieved this level of success when it comes to human brain. So we still don't know how the brain functions, which allow us to do many things that are uniquely human. Nor do we have answers to the majority of neurological diseases and disorders. So it is against this backdrop that I think that it is imperative for me to discuss with you the promise of neuroscience. So everyone knows that the brain governs learning, memory, consciousness, and many other higher order functions. And the nervous system is also very important in controlling vital functions such as breathing, heartbeat, sensing temperature, and sensing the pain. So you may or may not know that there are actually more than 1,000 diseases and disorders that are relevant to the nervous system. And these diseases really affect people across all ages. For instance, they include early developmental and cognitive disorders such as autism, attention deficits, and visual retardation, and can also include conditions that often develop in midlife, such as depression, anxiety, addiction, and other neurological conditions such as paralysis and traumatic brain injury. Finally, they also include diseases of aging, such as macular degeneration physician's information, mental and behavioral disorders and neurological and sense organ conditions, they are among the leading causes of use loss to disability worldwide in the year 2000 and again in the year of 2011, a whole decade later. So really, neurological and mental conditions can affect people across the globe. And among all of these conditions, I think the most terrifying disease is dementia. So if you talk to people who already reached middle life, if you ask them what they worry about, I think many of them will tell you that they worry about being forgetful and eventually develop dementia. I think this has become more concerning than even cancer or heart attack. Right now, it is estimated that there are about 35 to 36 million people living with dementia. And the number is rapidly rising. And currently, sadly, there is absolutely no effective treatment for dementia. And we can only watch the people being affected by dementia without any effective treatment. Similarly, in the Southeast Asia, according to a report from the International Alzheimer's Association, they did a survey in 15 countries, including Taiwan, in Southeast Asia. And I think the numbers, again, is very, very grim. And they estimate that in the next 2 to 3 decades, the people affected by dementia will likely quadruple. So they basically declared that 亚太地区时代这个已形成流行病 it becomes pandemic. So because the people affected by dementia is rapidly rising, and because it is such a difficult problem to deal with, in the last GA summit meeting, the countries agreed that dementia has become a global problem. And therefore, they decided to form the World Dementia Council, and the council met for the first time a couple months ago in London. These participating countries agreed to work together to build an international effort and to increase the funding for dementia research. And they set a very, very ambitious goal, which is to identify a cure or disease-modifying therapy for dementia by the year of 2025. So we're talking about 10 years from now that there has to be some therapy that can be beneficial. So among all the causes of dementia, Alzheimer's disease represents about 70 to 80% of all cases. Right now in the US, about 5.5 million people have Alzheimer's. And I think a very striking fact about Alzheimer's disease is that Alzheimer's-related death has risen by about 50%, while all other major causes of death, including cancer, heart attack, stroke, and even HIV infection, are on the decline. So currently, it is the 6th leading cause of death. And the disease is extremely costly because people with Alzheimer's disease are ultimately disabled. They cannot even care for themselves. They cannot recognize themselves. They cannot recognize their children or their close friends. So they have to be under long-term care, and most likely people would be residing in the nursing home full-time. Also, it is possible that family members will have to quit their job in order to care for the loved ones. So the annual cost in the US right now is estimated to be about $170 billion. But with the likelihood of the number of people affected by Alzheimer's triple by the year of 2050, the cost is estimated 1 trillion a year. So you can see this kind of cost. There is no country on the planet that can afford. And if we don't come up with an effective treatment quickly in the near future, I think the disease is going to change the structure and the economic situation of the entire world. So I think that Alzheimer's disease is not only a problem, it's not only a science and a health problem, but it is also a socio-economical and even a humanity problem. So the pathology of Alzheimer's disease, there are 3 invariant features. First of all, people with Alzheimer's lose a large number of their brain cells or neurons. So this results in a huge reduction of the volume of the brain. And if you look at the brain under the microscope, you will see 2 very salient pathological features. These are amyloid plaques, which are made of the aggregated and insoluble beta amyloid peptides. So deposition of these peptides precipitates in the formation of amyloid plaques. And there are also very abundant neurofibrillary tangles. And the tangles are composed of the aggregated intracellular protein known as tau. So because of these 2 pathological features, in the last several decades, people either in basic research or in translational research, they consider the plaques and tangles are the cause for the development of Alzheimer's disease, both the symptom and characterization, and they have been developing approaches to reduce the amount of beta amyloid peptides and tau protein produced in neurons in the brain. So, so in the past 2 decades, from the preclinical research all the way to phase 1 clinical trial, phase 2, all the way to phase 3, they have been many different approaches being created to reduce beta amyloid and tau. And after billions of dollars and tremendous manpower and effort, in the last several years, we only get to witness that every single trial, when it reached phase 3, they all failed. Okay, so there are more than a dozen of very high profile and costly phase 3 clinical trial failure. And in fact, not only that, none of this treatment improved Alzheimer's disease. A couple of them, in fact, made the patients worse. So this is really the whole process of identifying therapeutic treatment for Alzheimer's disease has been an extremely humbling experience for people working on Alzheimer's disease. So currently, there are a lot of rethinking and deliberation by researchers in Alzheimer's. And it can be summarized as the following to explain why none of the treatment has worked so far. So some people suggest that it may be too late in the game to treat patients already diagnosed with Alzheimer's. So in other words, patients need to be treated decades before the onset of memory decline. And in fact, currently some of the largest clinical trials supported by NIH and in collaboration between the NIH and big pharmaceutical companies are exactly doing this kind of preventive trial to identify people when they are in their 20s and 30s who are likely to have Alzheimer's disease to start the amyloid beta reduction therapy. And also, because some of the treatment actually render people worse. So people are also rethinking about the underlying pathophysiology of the disease. Is it in a relatively short period of time, large number of genetic risk factors have been identified. And the most famous one is the ApoE or ApoE4 allele, which can increase the risk for Alzheimer's disease by several fold. But unfortunately, because of the attention being paid to the plaques and tangles, very little is known about the contribution of these various risk factors to the manifestation of the disease. So these risk factors may provide a rich source of targets for new generation of therapeutics. And also another very interesting idea is that right now people talk about Alzheimer's disease as if it's a single disease, okay? But it is entirely possible that Alzheimer's could be a group of diseases, even though patients share similar symptoms and pathological features, but the etiology can be very different. And therefore, these patients may require very different ways to be treated. Finally, the brain is really composed of neural circuits. So there are billions of neurons and trillions of so-called synapses that connect each individual neurons. So the brain circuits that are most affected by Alzheimer's disease currently is still not known. And if we have this information, then perhaps we can come up with some approaches that we don't have to actually treat the entire individual or even the entire brain, but we can just focus on this particular circuit and try to reverse the pathology and symptoms. So because of this setback on the identification of therapeutic, the best way to deal with Alzheimer's disease, and because the disease is such a real threat, especially for the next few decades. Recently, President Barack Obama of the United States, he declared a national plan to address Alzheimer's disease. So I'll just show a short clip of the speech that he gave when he
You help
made
lead
this
the fight
announcement.
every day on behalf of the millions of Americans who suffer from Alzheimer's and the families who have to watch heartbroken as a loved one slowly slips away. It's incredibly important work, and it's only going to become more important as the baby boom generation gets older and even more Americans are put at risk. That's why my administration is committed to doing our part to help. As many of you know, earlier this year I signed into law the Bipartisan National Alzheimer's Project Act to bring together experts from the government and the private sector and create a plan to support those suffering the disease as well as the researchers working to find a cure.
So how do we really fundamentally improve what we are doing currently to better understand the brain function and to understand the mechanisms of Alzheimer's disease in order to find new targets for treatment? The Europeans, they really don't want to fall behind, and they already started the so-called Human Brain Project. This is supported by EU, and they already committed 1 billion euros to support this brain project. So this is gonna spend over a 10 years period. And the major focus for the European Brain Project is to collect all the existing data generated either from neuroscience research or systems biology research, or even from large-scale brain scans, images such as functional MRI and the PET scan. And then they're going to use supercomputing power to annotate this data and to figure out and to simulate the human brain function and to figure out what went wrong in the various neurological disorders, and through this to find better drug targets. And in the States, the Obama administration announced the so-called BRAIN Initiative, or known as Brain Research through Advancing Innovative Neurotechnologies. So this initiative was announced in 2013. And right now, the available information indicate that this brain initiative is going to be funded as a joint venture of the several federal agencies along with some of the private sectors. And the major objectives of the brain initiative is first to map the entire brain circuitry in human brain. And then they also want to measure the fluctuation of the chemical and electrical flow within the circuits. And finally, they want to understand the interplay between the two that creates the cognitive as well as behavioral capabilities of human beings. So in the next 10 years or so, the funding is going to gradually ramp up, and the major amount of funding is going to go into neuroscience, and particularly neurotechnology invention. So in the remainder of my talk, I would just like to go over some of the very exciting new technologies that have been invented that have greatly helped neuroscience and neurological disease research. So first, I would like to mention the induced pluripotent stem cell technology. This was invented by Dr. Yamanaka, who received the Nobel Prize for medicine in 2012. So the technology is very powerful because right now it is possible to take skin fibroblast sample from individuals who are healthy or who have different diseases. One can then reprogram these fibroblasts into pluripotent stem cells, or known as iPS cells, and then one can differentiate iPS cells into different cell types. And in this case, we can basically reconstitute all the different cells that populate the brain, including the different neuronal cell types, dopamine neurons, hippocampal neurons, and so on, and also the cells that support neurons, including astrocytes and oligodendrocytes. So in the Petri dishes, one can compare the cellular phenotypes between the healthy neurons versus the neurons from people suffering from diseases. And we can further make connectivity map and reconstitute brain circuitry in these Petri dishes and really figure out what is wrong with people who have the disease compared to healthy control. And finally, I think these cells and the system are going to provide the best model for drug screening. So you can imagine that the drug can be tested in the patient-derived neurons before the drug actually is eventually tested in the patients. And people continue to improve the iPS technology. So now the technology can go from the two-dimensional culture to go all the way to become a three-dimensional culture. So this was first reported by end of last year from a group in Vienna. Now they first use iPS cells to make a droplet, and then they can use the bioincubator to provide flow to the droplets and enable the droplet to develop eventually into cerebral organoids, so like a mini brain. So if you look at this image, I would think that this is actually an image from a fetal human brain, but in fact, this is something that's made out of initially the individual iPS cells. So we can further make the iPS model to be much more sophisticated and much more brain-like, which would likely to further provide information about how the brain works, what goes wrong in disease, and serve as a very, very good model for testing potential therapies. Another technology that has excited the scientific community is the ability to rapidly edit the genome. This is known as the genome engineering technology. So before this, it was possible to edit the genome, but it's very labor-intensive, very tedious, very costly, and very time-consuming. Now, with the new way of using the CRISPR-Cas system, one can literally edit the genome of any mammalian cells within a couple of weeks. So the technology was invented by Fan Zhang at MIT and George Church at Harvard Medical School. They use this Cas9 nucleus, which can be guided by a small guided RNA to any loci in the genome, and then the nucleus would make cut or double-stranded break in the genome and completely get rid of a chunk of the DNA or replace that part of the DNA with any sequences of your interest. So why is this powerful? Let me give you an example. So Down syndrome, I think many of you are familiar with it. People have Down syndrome, have intellectual disability, and early onset of Alzheimer's disease. And the disease is caused by trisomy 21, or 3 copies of chromosome 21, as opposed to, for all of us, we all have 2 copies of every chromosome, except for the sex chromosome. So what has been done today is the fact that people can take Down syndrome patients' fibroblasts and then reprogram them into the iPS cells. And the cells will have the 1 extra copy of chromosome 21. Now, one can use the genome engineering technology to introduce this so-called exist gene into the extra copy of chromosome 21. So what this DNA would do is to encode a particular kind of RNA that would spread the entire chromosome and completely silence this entire chromosome. So the resulting cell, they exhibit property as if they only have 2 copies of chromosome 21. So I think this observation is extremely exciting because it opens the door for potential gene therapy and regenerative medicine. Now, when you look at this image, you would think that I'm showing you an image with 1 mouse brain. But you may be surprised that on this slide, there are actually 2 mouse brains, okay? So there's a mouse brain here, and there's another mouse brain here. Except for that, this brain is completely transparent because it has been processed by this so-called clarity technology. And the technology was invented by Kwang Kun Chan and Carl Diceras. And Kwang Kun now is an assistant professor at MIT. So what they did was to replace the brain's lipid with this hydrogel biopolymer. Okay? So the hydrogel biopolymer can deflect the light in such an angle that renders the brain completely transparent. So you may ask me, what is it so good to have a complete transparent brain? So you know, right now, one of the biggest tasks for the Obama Brain Initiative is to map the entire mammalian brain circuits and eventually human brain circuits. But it is really not very easy. To do that, currently the best thing one can do is to thinly section the brain into this kind of thin section that can be just micron thin. And then you stain the different sections with antibodies against different cell types, against different axonal tracts, and then you acquire the images 1 section by section, and then you're going to use some sort of computational power and algorithm to reconstruct the 3-dimensional brain, okay? And it's going to take many, many, many years to get this done, even for a mouse brain, let alone a human brain. But with now a completely transparent brain and with enhanced and improved microscopy, one can actually visualize the entire neural circuits in its entirety. So let me just give you an example. Here, the excitatory neurons of the entire mouse brain is labeled with the green fluorescent protein, okay? So now you can see, you can actually visualize the dendrites, you can have the single cell resolution of all the excitatory neurons. You can also see this entire projection, the axonal projection, from one anatomical location to another. Okay. And this brain actually can accommodate antibody staining for 30, 40, 50 different markers. So you can further appreciate the interactions among all of the different cells. Another technology I would like to introduce to you is known as optogenetics. Okay. So what does that mean? This is a technology allow you to activate neurons, to control neuronal activity in a millisecond time scale. So how do you do that? This largely is made possible through the identification of light sensing channels from initially algae. So I just want to give you a couple of examples. CHR2 is known as channelrhodopsin 2. It can specifically be activated by the 470 nanometer blue light. So if you introduce channelrhodopsin 2 into neurons and then expose the neuron with this blue light, then you can activate the activity of neuron in millisecond temporal resolution. MPHR, on the other hand, is known as halorhodopsin, and it can be specifically activated by the 589 nanometer yellow light. And when neuron has MPHR and then be activated, and the channel being activated, it will result in a complete silencing of neuronal activity. So now you can use genetic method to introduce these channels into different neurons in different anatomical positions, and then activate or inactivate these neurons and evaluate the consequences in either our behavior or electrophysiological properties of the cells. So I want to show you a very short movie created in my lab where we introduce channelrhodopsin 2 into the excitatory neurons, specifically in the basolateral amygdala, which is a brain region controls valence and fear behavior. Okay, so you can see the mice have fiber optic implanted, so we can introduce the light, and the mice normally love to explore the environment. But once we turn on the light, you see the mouse completely change the behavior. Now it's back to the corner and sort of hunched, its back is hunched. It sort of shows this ultimate fear response. But if we turn the blue light off, within a few seconds, the mouse will resume its normal, active, happy kind of exploration behavior. Okay? So through this kind of technology, you can go brain region by brain region, neuronal type by neuronal type, to really understand which cell and which brain region is responsible for what kind of behavior. So my last slide, I just want to say a few words about the amazing revolution of our ability to sequence the genomic DNA. It doesn't seem that long ago when the first draft of the human genome sequence was assembled. It was in 2001, and it took a long time, many years, for this to be completed, and it costed $40 million. But then with the invention of the so-called next generation DNA sequencing technology, in 2007, the first human genome was sequenced by this new technology. But back then, it was very, very expensive. It costed more than $100 million to get this first human genome sequenced. But then 2 years later, look at this. The price literally dropped by about 100-fold. So the first Korean genome was sequenced with the cost of only $1.5 million. And then the price further dropped by the order of magnitude. So today, to sequence a complete human genome will only cost about $2,000. And the price is still dropping. Okay? So what it means is that basically everybody can have their genome completely sequenced. So we officially transitioned from the so-called Human Genome Project to the Personal Genome Project. So why is this helpful?
So when you think about Alzheimer's disease, I told you earlier that so far none of the clinical trial worked, okay? And I also brought to you this possibility that Alzheimer's disease may be a group of many different kinds of disease, and there is this need for the stratification of patient population. And this is actually happening right now. So for the next generation clinical trial, for instance, currently a leading pharmaceutical company recruiting patients for phase 3 clinical trial, they already signed up about 100,000 people. And they also obtained the consent of every single participant to allow their genome to be sequenced. So I think this company now is in a very good position, because regardless of the outcome of this trial, they're gonna learn a lot about the response of this individual to this particular treatment. And they can see perhaps there is a correlation between their genotype versus their response to this particular treatment. Okay, so this is gonna allow them to further the approach to come up with a better medicine for different population of patients. So, so in summary, I would like to just give you some positive perspective for the future of therapeutic intervention of Alzheimer's disease. I think that with all the new technologies that have been invented and the new ones are still coming up, I think we can be optimistic about what's going to happen in the future. I believe that there will be novel therapeutic targets and drugs that can be very much better validated initially in patients' own 2-dimensional or 3-dimensional brain model system to be tested before they actually go on to the patient themselves. And the personalized genome now can allow for stratification of patients and further can facilitate personalized medicine. And with the stem cell technology and genome editing technology, one can also think about the very realistic possibility of regenerative medicine. And brain stimulation techniques now, I think, provide a real hope for the fact that perhaps for some diseases, you don't need medicine, but rather using electrical or other means of stimulation of a particular brain circuits will be much more effective and safe. So for the future, I think we will come up with treatments that would make a difference. And I thank you very much for your attention.
谢谢蔡院士给我们很精彩的演讲，他把最新的在21世纪的新的这些 biotechnology 用来做 Alzheimer diseases 的研究，就如同他讲的一样，到未来呢，我们可能有一个 personalized 的medicine regenerative medicine， 或者甚至不要用药物，而是用一个电极，就可以来解决人类的这个问题哈。那科学就是一个不断的接受挑战，不断的去为人类的福祉，追求更好的治疗的方法。我们是不是再次谢谢蔡院士精彩的演讲？